So this slide is about our genetic factors. First, I want to mention that medullary thyroid cancer is 5 to 8% of thyroid cancers, so it's really not that common. And then out of those 5%, 70 to 80 are sporadic cases. And then what we'll be talking about, hereditary, is 20 to 30%. And the genes involved in hereditary are transmitted as autosomal dominant, meaning that an offspring has a 50% chance of receiving it. And then that predisposition gene is RET, a proto-oncogene that's involved in normal cell growth. And they're characterized by constitutive ligand independent tyrosine kinase, which is a nonstop functional state that can contribute to initiation or progression of cancer. And then the RET receptor plays a role in regulating cell division, migration, and differentiation. A little bit more about the RET gene is that it is 10q11.2, and then it also has 21 exons. And the two of those exons, 10 and 11, are germline mutations that are responsible for the development of cytopathology of the familial medullary thyroid cancer. And then the RET protein has three parts. There's the extracellular, which is the cytosine rich domain, and that's two mutated RET molecules that combine and activate. There's also the transmembrane, which ensures close proximity to the RET proto-oncogene monomers through non-covalent receptor interactions. And then last is the intracellular region, which is juncta membrane domain, and it also has two tyrosine kinase subdomains, TK1 and TK2, that are involved in the activation of intracellular signal transduction pathways. And then mutations, the mutation originates in the thyroid C cells that make calcitonin, and that's a hormone that helps with calcium level in the blood. And mutations are responsible, the mutations responsible are missense mutations, specifically gain of function, and that results in the protein being all wonky because of the changes in amino acid sequence. There's three types of mutations, one a subdivision of ME2A, and that is just patients develop multifocal bilateral MTC, which is normally 100% penetrance. The pheochromocytoma, which is 42% penetrance, and then the hyperparathyroidism, which is 10 to 30% penetrance. And then 98% of the ME2A patients, mutations occur in one of the 5 cytosine, extracellular cytosine rich domain of the RET. And then the subdivision for MEN2A is FMTC, or familial medullary thyroid cancer. And that actually has no clinical feature. And then those mutations affect either extracellular cytosine or intracellular domain of RET concerning all previous exons. And then the second one is the ME2B, and that's characterized by specific clinical phenotype with ganglion neuromas of the lips, tongue, conjunctiva, musculoskeletal abnormalities, as well as a long narrow face, among other features. And then 95% of those cases carry a mutation on the RET exon 16. And then our SNPs, or SNPs, are single nucleotide polymorphisms. That is our SNP G691S in exon 11 and SNP S904S on exon 15. And they're responsible for the development of medullary thyroid cancer in patients with germline RET proto-oncogene mutations, and also believed to influence age of onset. And then the highest risk factor is the classic MEN2B on M918T carriers. A high risk is patients with RET codon C634 mutations, and also a moderate risk includes patients with hereditary medullary thyroid cancer and RET codon mutations other than the ones I mentioned previous.